go ahead and get started. I'd like to introduce Dr. Matt Jolly. He's an ecologist with the Fire, Fuel, and Smoke Science program at the Missoula Fire Sciences Lab. I've gotten to know him over the years because I'm on a Northern Rockies incident management team and have had the opportunity, the good fortune to work with Matt on a number of different occasions. He is a PhD, having received that from the School of Forestry at the University of Montana. And the topic of interest for him is live fuel moisture. He says it's measured frequently throughout the country as an indicator of potential fire behavior, but is little known as a primary factor that drives seasonal variations. He'll delve into the interactive factors that control live fuel moisture, and we'll discuss some of the potential implications of these factors on seasonal variations in the fire potential of living plants. Ultimately, he'll show how the interactions between the water content of the foliage and the seasonal changes in the leaf's dry weight combine to influence the calculated live fuel moisture and ultimately its flammability. So with that, Matt, I'll turn it over to you, and we will certainly entertain questions. If something seems appropriate at that time, I will ask if we can stop and answer it directly. Other than that, we'll find the right time to ask questions, and there will be a period for Q&A at the end. Matt?
Awesome. Well, thanks, Zeke. I'd also like to just thank both of the consortiums for having the chance to come out and talk today. These webinars are oftentimes challenging because you can't see people's faces and you don't get that person-to-person interaction. But I do encourage everyone that if you have questions as we go throughout this talk, to use the chat feature. We'll stop and answer those questions as we go, or yeah, there's actually a little hand raiser up there if you want to use that as well. So I recommend that we do that as we go. And I'd also like to just start out by thanking collaborators in addition, because you can never do anything by yourself anymore. So we've had lots of good help on many facets of this project. Ann Hadlow and Kathleen Huguet and Rachel Kropp. Sarah McAllister is a research mechanical engineer here, and Mark Finney. So we've all been really trying to sort out the different pieces of this puzzle, and we'll talk about how that works. So from my perspective, and my motivation has always been that that we understand that fire in the world is common, and particularly fires in live fuels are generally the ones that are the largest and that burn the greatest extent. And we understand that that on one side we have this thing where fire is bad, essentially that we know that it poses the greatest risk to life and property, and that fire and live fuels particularly are resistant to control efforts. But on the other side, we know that fire is fills a valuable ecological role. And really what my main focus is to try to figure out how to balance those things by developing more understanding of how all these important factors come in to influence flammability, so that we can provide tools to land managers to actually be able to assess what kind of fire they can expect at certain times of the year, and how those things add up to influence the fire potential. But what you realize, that particularly working here at the fire lab and around in this fire behavior science realm, is that our history quite often shapes our perceptions of how we address a problem. So one person that's been very influential in fire behavior research is Byram, and in 1959 he wrote that the effect of fuel moisture on the burning rate of wood is pronounced and so obvious that no measurements are needed to illustrate its overall effect. And I think that this quote is really illustrative of the sort of fundamental underpinnings of what we do as fire scientists, because we're constantly trying to go back to fuel moisture being the key of what makes things burn, and we'll talk about why that is or isn't always the best approach, because these sort of statements lead us to that natural conclusion that basically when we talk about live fuels and how they burn, that live fuels are just really wet, dead fuels, and that from what we're learning is pretty much anything but the case. We know that the combustion of living plants is really we have several things that are happening as something is burning. A fire propagates by having a burning particle adjacent to an unburned particle. That unburned particle is heated. Some or all of the water is driven off. It's heated through radiant convective heat transfer. We know that combustion isn't always complete. We know that water vapor is driven off into the flame both during preheating and during the combustion process itself, and that basically oxygen is used to, things are oxidized to create the heat that helps the fire propagate. So it's we know that water plays an important role in how things burn. It's not, anyone that's tried to burn wet wood in a campfire understands that fundamentally. What we don't know is to what degree it influences how it influences fire propagation. So at the scale of a fire, Byron in 59 showed basically that the effective heat yield decreases as the fuel moisture increases. And these are just basically calculations from energy calculations from the fire that are incorporating things like the fire not being very efficient, and that basically as the fire increases, as the fuel moisture increases, the effective heat yield of the fire decreases, and that effective heat yield actually goes down as the size of the fire increases because the fire becomes less efficient. So we know that it takes energy to heat up and evaporate moisture, and that the role of water in the flame itself can have a dilution effect. So we're acknowledging that it does play some role. Well, based on our history, what we've done is develop an entire set of systems that are built off of using live fuel moisture, or whether it be from the canopy fuels of a conifer or the surface fuels of a shrub. All of those things are generally used as inputs to models. So this is just an example of a couple of different models that have been put out over the years, but there's many of them. So the figure on the left shows a fire spread calculation from Behave Plus using the Rothermel fire spread model for a surface fuel model, and over a range of live woody fuel moistures. So you see basically as the fuel moistures increase, the surface rate of spread decreases. These are all things that are intuitive to people because they've seen these tools before. The one on the right of this page is basically a probabilistic model for chaparral that says what the likelihood of a fire propagating through chaparral is under a certain set of conditions, and it's just the results of a logistic model that was developed by David Weiss and his cooperators a few years back from laboratory burns, correlating laboratory burns to whether or not the fire propagated. And then we have lots of lab studies as well. So the lab studies go into in-depth into looking at how they go in-depth into looking at things like the time to ignition. So basically you heat something up under a fixed set of conditions and you measure how long it takes to ignite. And that ignitability is related to one of the factors of how flammable they are. So all this is really meant to point to the fact that that we start out with this original frame of saying that fuel moisture must be the main thing that we're looking at, so, because we do understand that in dead fuels it is very important, and the effects are quite easy to show and have been shown in study after study. But we've tried to extend our thinking from the dead fuel realm, where most of the original models were developed and most of the lab studies are done, up to this these live fuels, to try to say that live fuels are basically just really wet dead fuels. But I still think that there's things that we don't understand, and it's very unusual that a fire will spread. So in dead fuels, we know that fires generally won't spread when the fuel moisture is greater than about 20%. It varies by fuel type, and this is usually referred to as the moisture of extinction. But we know that for live fuels, it's not uncommon for a live fuel to burn when its moisture content is at 100%, so, or 90 or 80 or whatever. But the important thing is that we have a fuel that's fuel moisture is 5 times higher than a fire would typically propagate through a dead fuel, so they must be different in some way. The one thing that we really don't understand at all are the interrelationships between all of the things that are happening inside the plant and how those things are affecting what we calculate as its moisture content, what its thermal and physical properties are, and then also how it burns. Because what we typically do in science is we look for that smoking gun, that one factor or that one variable that explains everything. And I really feel like that when it comes to the combustion of live fuels, we're not going to find that, because everything is connected, and that's what I'll show you as we go down. One of the things is we spend a considerable amount of time and money to measure live fuel moisture in the field. We measure it, document it, and but people are always asking me, what the utility of these measurements are, and sometimes I just don't have a very good answer for them, because a lot of people actually try to interpret the live fuel moisture changes as changes in essentially the drought stress of a living plant. And I'll show you a little later why that's really not going to work. So what we need is a way to really help understand when you are measuring the fuel moistures. One, how do you interpret them? What's really happening inside the plant, and then two, also to see if there are other things that we could measure along the way that would be more related to the seasonal changes in the flammability. And all this really just assumes that we know what live fuel moisture is quantifying. So we've started out from that original discussion of Byram saying that fuel moisture is so pronounced that we don't even need to measure its effects. And then we built models around it. So surface fire spread models, the crown fire models, lots of models use foliar moisture or live surface fuel moisture as inputs to their models. But we really maybe don't even know what that live fuel moisture value that we're measuring is actually quantifying. And the big difference lies in the fact of, if you look at live and dead fuels. So if we think about a live fuel, that say a pine needle that's laying on the surface, and we go and pick it up and we put it in a can and we take it and dry it and weigh it to get its fuel moisture content. Well, the one thing that we can be certain of is that over the time of a fire season, for example, the dry weight of that sample is most likely not changing very much. So our equation down in the bottom here for live fuel moisture is the same equation for any kind of fuel moisture. It's basically just the fresh weight minus the dry weight over the dry weight. So we do understand that that for a dead fuel, that dry weight, it remains constant. Well, the real rub happens when you start thinking about a living plant or a live needle. we know that the fresh weight of a live needle changes because of things that we'll talk about in just a second. But there's also the potential that the dry weight of the needle is changing as well. So when you're actually calculating a live fuel moisture, you don't really know, what the main drivers are of the seasonal changes in that calculated moisture content. So we've always kind of assumed that that live plants and dead plants or live fuels and dead fuels are the same, but the simple fact that the dry weight of a live fuel can change over time complicates this picture greatly. So if we think about it a little bit differently, for a live fuel, basically we have two separate pieces that are happening in concert to affect the calculated live fuel moisture. The side on the left looks at the fresh weight changes, so the actual water content of the plant. And we know that those things that the actual water content of the plant is driven by the water balance of the plant. So things like the water uptake and storage from the soil and then the stem, and then water loss through transpiration. And then on the right side, though, we have the things that affect the dry weight of needles. We know that something as simple as photosynthesis, if it's happening rapidly, can accumulate carbon in the needle. That accumulation of carbon can actually increase that needle's dry weight, and if you increase the dry weight without actually changing the fresh weight at all, you can change the calculated live fuel moisture. So in our example, if we increase the dry weight by accumulating carbon, we would the result would be a lowered or decreased live fuel moisture that we've calculated, even if the fresh weight stays the same. One of the big things that probably drives the seasonality more than anything is the phenology. So we know that, and I'll show you some examples from our field measurements, that new needles most conifers can have moisture contents about 3 times higher than that of the old needles on the same branch. So it makes it very, very challenging to try to figure out how to put all these pieces together to really sort through the dynamics of what's going on. So our goal basically is to determine the factors that control live fuel moisture and assess how they might affect their flammability. But the most important thing is also to look at the linkages between live fuel moisture and all the other things that we know that are important in terms of flammability for the plant. So what we've done over the last few years is actually collected foliar moisture content samples. We've focused mainly on lodgepole pine because, well, we started sampling lodgepole pine, and then it's a relatively straightforward thing to sample here, and we found that most of the results are extended to other conifer species quite readily. And we've also done quite a number of things that we won't present on broadleaves, and they all look the same. So the key, though, is that we have a few years of these samples now at several sites. But one of the things that we did when we were sampling the foliage is we separated the new and the old needles. So we calculated the oven-dried live fuel moisture and also the relative water content for these samples through time. The relative water content is actually very similar to live fuel moisture content, except that it's expressed, the number is expressed relative to the amount of water that the foliage can hold. So it's always naturally bounded between 0, which is it has no water in it, and 100, which means that it's fully saturated. And we also did a few other things by looking at needle density, which we found actually to be quite important and might actually explain a lot of things. And then also we've been looking at the seasonal changes in the heat content of the foliage. Another thing that we did is for those same samples, we sent them off to an external lab for chemical analysis. And the chemical analysis that we use is a forage analysis that breaks the breaks the chemistry of a particular needle up into these nice big broad categories that we can use to see how the needles are changing over time and which category is increasing or decreasing, and hopefully to correlate that back to how the dry weight is changing. So, and then finally we we actually ignited samples a couple of different ways. We have a radiant panel setup, which we'll present later, and then an open flame burner. The open flame burner is as close to a wildland fire flame as we can get. It's a very, very rapid heating flame, and we actually have attached to that a nitrogen quenching chamber, which is really pretty neat because we can actually take a fuel particle, introduce it to the flame, heat it up to just to the point of ignition where we get flames. We can pull it back into that nitrogen quenching chamber and snuff it out. So we actually have a sample that was heated up just to ignition, and we can see how that's changed. And I'll show you some preliminary results of the moisture loss at ignition, which is going to be pretty important as well. So for the fuel chemistry, we have the forage analysis that we get actually is, as I said, it breaks it up into these nice big groups of chemical compounds that make up the foliage. So our needle dry matter is broken up into its percentages of these groups. So the first one is protein, which is mainly the photosynthetic components of the foliage, and then crude fat, which is a large category, includes everything from the waxes and on the cuticle to terpenoids and pretty much anything fat-based, and the ash content, or the inorganic or mineral content, which is pretty standard for a lot of this stuff. But the two other things that are really interesting is the non-fiber carbohydrates, which are also broken out into starches and sugars. So basically the simple byproducts of photosynthesis and or energy storage in the plant. And then on the right hand side, it actually also tells us about the structure of the actual structure of the plant or the needle. So that structure is we know that they're made up of cellulose, hemicellulose, and lignin, and this test will actually separate out for a lot of that stuff as well. So right away we start seeing differences even within the same plant and the same foliar category throughout a season. So the graph on the left, these two pie charts are actually both for new lodgepole pine needles at the beginning and end of the season. And the great thing about this chemical analysis, along with the moisture content, is we can actually look at everything that the fuel is made up of at that time. So in this case, we know that the needle is actually about 65% water for a given kilogram of fresh fuel, and surprisingly about 30% of carbohydrates, and a small percentage of crude fat, crude protein, and ash. So, but at the end of the season, as the moisture content declines and the needle hardens, we see that the needle is actually made up of a lot less water, so about 12% less water, but also quite a lot more carbohydrates and a higher percentage of crude fat, roughly the same crude protein, and the same ash content. So it starts to point at the things that are varying seasonally in the foliage. One of the things that I find really important, though, is that okay, we have a question. I think that's the first question. I hope I haven't missed any here. So Persephone is asking, what do we mean by season? And by season, we're looking at, we sample throughout the growing season from green up to basically till we can't get into the sites anymore. We actually sample from snow off to snow on. So season would be, in the case of the previous slide, it would be the beginning of the season, meaning that right at green up, and the end of the season, meaning once the needles have hardened.
And Matt, that was the first question.
Okay. Don't let me go so fast that I miss any. Gary, crude.
So, yeah, Gary just asked about the crude fat that you talk about.
So the crude fat is also what everybody refers to as ether extractives. So the ether extractives are pretty much, well, anything that's any fat compound in the needle. So they call it crude because it literally is everything that is fat based in the needle. So things like the waxes and on the outside of the leaf, all the terpenes are also coming out as crude fat, and even some of the cell wall components.
And just a follow-up question to that, Matt. Which of these chemical compounds are the ones that are most flammable in the living plant?
So we know that, and I'll get into that, but we know that crude fat, well, fat in general has twice the heat content of any other component of the plant. So the crude fats are definitely more flammable, and also, but I'll show you that it's actually in here if we can table that question. But hang on to it if I don't answer it. Gary, I think you're right. So Gary asked if the external cutin or the wax buildup on the outside of the needle could possibly inhibit ignition. And I actually believe that that's possible. What we found that is not in this talk, but it's very interesting, is that we can have live needles. So a new needle and an old needle. The new needle doesn't have a cuticle that's built up on the outside of the needle yet. It's not protected, and that's essentially what happens as it develops and hardens. And we can have an old needle just right next to it. We can burn them at the same time, and despite the new needle having a moisture content that's almost twice or sometimes 3 times higher than the old needle, that new needle will actually ignite faster. And I still can't explain why, but I actually believe that it's because you have to burn through the cuticle before you can drive the water out of the needle. So I do believe it's possible that external cuticle can affect the. Cool. Well, please ask questions as we go. So one of the things did I get your questions, Gary? All right. So I think that one of the things that almost every fire model does is assume that live fuels are just like wood. We use the combustion parameters for wood for almost everything. Well, what we see from this graph is that if we look in the upper left-hand corner of what how a pine needle's dry weight is partitioned, we can see that 41% of it is actually structural carbohydrates, so cellulose, hemicellulose, and lignin, but 42.9% in this example is made up of non-structural carbohydrates, which are basically simple sugars and starches, which is pretty amazing because wood generally is just cellulose, hemicellulose, and lignin, with very, very minor contributions of other parts. So right away we see that the live needles are chemically different from wood, but we also see that in the center, in the lower part, that there's a large proportion. We're going to try this drawing tool out. There's a large proportion of a dead needle that's in structural carbohydrates. So, and a much smaller proportion that's in non-structural carbohydrates. So, if you just look at these graphs, you start to get a sense that hey, one of the things we've overlooked all along is that live fuels are very, very much chemically different than wood, but we still seem to use wood as our reference for everything. In the upper right, you'll see the same analysis for a Douglas fir, and you'll see that in this that for these samples, 55.7% of the dry weight is in non-structural carbohydrates, so sugars and starches, and a fair amount of it in crude fat on both occasions. So it's pretty important to keep that in mind. So here's the results of a field sampling for live fuel moisture for a year. So we started in May and sampled through to November. But what you see is it illustrates a few things. One, it illustrates the importance of separating new and old needles when you sample live fuels. If you look, what you see for the new needles, the sort of red and or darker lines, is that they start out really high, but they decline throughout the season until by the end of the season out here, they're roughly equal to each other. So the new needles and old needle moisture contents pretty much are the same by the time those needles harden. What you notice with the old needles is almost no variation at all compared to the new needles, but it's you actually see an increase in the old needle moisture content as the new needle moisture content is decreasing. Well, what happens during needle development is that the new needles are a net sink of carbon to the plant, which means that they cannot possibly produce enough carbon to maintain their own growth and maintenance respiration. So the only way that they grow is for the old needles to give carbon to the new needles. So it's that translocation of carbon out of the old needles and into the new needles is what makes these two curves very different. So if you look really close in here, and we have better examples from other data, but it makes the old needle moisture content look like it's increasing because it's taking carbon out of the old needles, which is making the old needles dry weight go down, and its moisture content, calculated moisture content, look like it's going up. And for the new needles are actually getting heavier. So they're receiving that carbon from the old needles, and they're elongating, they're developing that waxy cuticle. I think we have another question. I'll come to your question a little later, Ron, if that's okay. I promise I'll get to it.
Yeah, we'll keep track.
So basically, what we get is a really interesting dynamic, and that is that the new needle moisture content the dynamics of the new needle moisture content are driven by that needle getting heavier because it's receiving carbon from the old needles. But what happens when people sample a lot is they don't separate new and old needles. So they're mixing in two very, very different populations into their sample and hoping that they have a representative sample. So from a sampling perspective, it's very important. A lot of people just focus on sampling the old needles, but then you miss the fact that the tree could be, some portion of the tree could be at a very, very high moisture content. So one of the interesting things about the relative water content measurements, though, is that they're very different. If you look at them, they actually don't vary very much over the season, but what they do show some pretty interesting things, I think, and one of them is this dip. And that dip happens during the period of the lowest seasonal relative humidity. So it's actually picking up the actual water stress of the plant better than live fuel moisture content is. And physiologists use this metric all the time for water, any kind of water-related studies in plants. And it's so it could be that we still need live fuel moisture content for many of the models, but using relative water content by itself could actually be the kind of metric that we're looking for to quantify the sort of drought stress of that plant. If we look at the needle development throughout the season, this is basically just a measurements of needle length over time, normalized to the maximum, and there's a little bit of noise on the upper end, but what you see is that it takes about 60 days for needles to reach full length or full maturity. So if you can imagine at the canopy level, you have this population of old needles with a relatively low moisture content of, say, somewhere between 90 to 120%, we found to be very normal. And then at the point of green up, you have a portion of that canopy that's actually at a very, very high moisture content. So even as much as we measured a Colorado blue spruce that had a 414% moisture content. So, but it might not be a very large proportion of the total crown biomass. So if we look at the sort of aggregate of those two things, we can show that our new needle moisture content starts out high as the green line and declines. Our old needle moisture content is increasing, so we're basically going kind of up like this and down like this. But if we use that previous graph to balance out the proportion of the canopy that's in old and new needles, what we see is a characteristic curve that everyone kind of expects to see, is this rise and increase to some maximum and then a decline as you approach the fire season. So it really is an interesting problem because there's so many non-standardized methods of how we actually measure fuel moisture out there that it's hard to compare all of these things. So one of the things that we look at, if we look at throughout a growing season, for example, how all the chemistry is changing, we see a couple of interesting things. Is one, for the non-fiber carbohydrates, which are basically the cellulose, hemicellulose, and lignin, they don't change that much. So they stay relatively constant percentages for both new and old needles throughout the season. What we see the most variations in actually are the starches and sugars. So you can actually see that the starch-sugar content increases, almost doubles for new needles throughout the season. So it's increasing like this, whereas the actual starch and sugar content of the old needles is going down. You can see this dip right at the point of green up, and then it stays relatively constant. And this is the point where it starts to give carbohydrates from the old needles into the new needles. Well, we see the same thing in crude fat, is that the crude fat of the old needles stays relatively constant throughout the season, but the crude fat of the new needles is actually tripling as those needles are developing over time after green up. So you'll notice that the shape of this of panel E and panel F looks very, very much like the shape of the decline in new needle moisture content, new needle foliar moisture content that we see up here in panel A, and the same for the old needle moisture content. So they're very highly correlated. If we look at it a little bit different way, if we plot basically the foliar moisture content against some of these key variables, we see that one of the nice things is that relative water content and foliar moisture content are related. It's not a very strong relationship, but they are related. So at least some of the changes in live fuel moisture are happening as a change in the actual water balance of the plant. But we see over here that that the non-structural carbohydrates, which are the starches and sugars, are almost perfectly correlated to the changes that we see in the live moisture content. In fact, with starch and sugar alone, we can explain about 82% of the variation in foliar moisture content without ever knowing how much water is in the needle or leaf. And the same with crude fat. So we see that a very, very strong relationship between these two. So if we look at the percentage of non-structural carbohydrates and fats as a percent of dry matter against live moisture content for lodgepole pine, we see that there's about we can explain 87% of the variation just with that combination of two variables alone. But if we look at it across many species, so this is the same thing. The black line is actually the same line from the previous graph. It's just extended out. But you'll see that if we plot Douglas fir out here, which it has a lot of starch, sugar, and crude fat, up to 45% of its dry weight. And we have western larch, Colorado blue spruce, ponderosa pine. All those things line up to tell us that just non-structural carbohydrates and fats can explain almost all the variation in live fuel moisture. So what we see as well is that that if we combine them in a simple linear model, just log transforming the data, that the combination of relative water content, non-structural carbohydrates, crude fat, and actually ash content turned out to be significant. We can explain 92% of the variation in live moisture content. So basically what we've done is kind of decompose it into the things that drive the water balance, which we use relative water content as a metric for, and the things that drive the seasonal dry weight changes, which are based on the non-structural carbohydrates and crude fat and ash. So one of the things is how does the chemistry actually affect the fuel's physical and thermal properties? And one of the things that it actually affects is, okay, we have a question from Walt Kessler. It says, how might a spring drought change the typical seasonal live fuel moisture picture? And I think that's really it's actually kind of a hard question, because we know that if a true drought drives the relative water content down, then that should drive the calculated live fuel moisture content down as well, if you're only looking at the changes in the water balance of the plant. But what happens is that if it's during drought, the plant actually closes down, doesn't photosynthesize, so the dry weight of the needle actually might go down as it's consuming the carbon that's fixed and not fixing new carbon. So it could actually be that the net result is no change. So if we talk about fuel density, though, and basically it's just the weight of the fuel normalized by its volume. One of the things that we typically focus on is that we use the density of wood for almost everything, and we've never actually tried to measure the density of a needle or a leaf to see if it's any different. Well, what we found from many foliar density measurements is that they're significantly different, and sometimes as much as 3 times less than the density of wood. The reason that's important is because the ignition of a particle is directly proportional to its density. So if you increase the density, you increase the time it takes to heat that fuel up and to change its temperature to the point that you get to an ignition temperature, that you're pyrolyzing the particle. So in this case, the median was 400, so it was about 20% less, but it went down to as little as 200. So if we look at seasonally, the bottom part of this graph shows the density of new needles, and the top part of the graph shows the density of old needles. And lo and behold, you'll see that they look very much like the opposite of the moisture content. So we have a physical property that we know is related to its ignition characteristics that's linked by its chemistry, because it's essentially getting harder as it's or heavier with its volume staying the same. But you'll notice that these aren't trivial differences either. That we start out at over over 0.6 grams per cubic centimeter here, whereas the density of a new needle is about 3. So we're talking about a literally 100% difference. So John said that the Wisconsin samples sampled during hot, dry 2012 spring and summer showed no difference in live fuel moisture trends from the previous normal years. And I think that goes back to your question, Walt, is that I really feel like that what we get into is sort of competing things that are going on within the fuel. So if you're changing the dry weight and you're changing the fresh weight, you can't really relate that back to drought per se. But if you were to try to do these relative water content throughout the season, which are actually just about as easy as doing the live fuel moisture content, you'd probably find that it would show those trends. So so if we go back to our density, what we see is basically the density literally has almost 100% variation depending on the season and the particle that you're sampling. Some of the other things that chemistry affects is the specific heat. So we know that actually from food science you can separate the specific heat into its respective parts, and what you see is a pretty pronounced variation when you include the water and the specific heat of the protein and the crude fat and carbohydrates and ash into this bulk specific heat measurement. You see that that basically in the springtime, new needles have a very high specific heat and old needles have a very low. But what that means is that the new needles can absorb more energy without changing their temperature. So it affects their flammability. And one of the things, and this kind of comes back to your question, Ron, is that our crude fat measurements actually quantify the things that are considered volatiles. So the volatiles or ether extractives come out as part of this crude fat analysis. And one of the interesting things with the crude fat is that it's really closely related to the heat content of the fuel. So this is just an example from a sagebrush that we did this from sagebrush heat content that we did this summer. But what you'll see is just even over this short period, as the sagebrush needles are developing, their heat content is actually increasing. And that is mainly related to a change in their crude fat or the volatile extractives or whatever you want to label them. So you'll see the same trend here, but more importantly, if we look at it over a range of species, we'll see that crude fat alone is a nearly perfect predictor of the heat content of the fuel. And the heat content of the fuel defines sort of the upper end of how much heat that fuel can generate if it's completely combusted. So this graph actually shows 3 points that were from Much, Bob Much, a paper by Bob Much in 1970, and then also shows our lodgepole pine data for the average crude fat that we got from our chemical analysis and the average heat content that we got from bomb calorimeter data, and then the same with sagebrush. So you can start to see how all these pieces interact and link together. So basically, if we go to look at the ignition, we've done many ignition tests here using a couple of different apparatuses. One of them is that the ignition apparatus that we use for the radiant heating is called a forced ignition flame spread test. It consists of a small wind tunnel. So this is basically a fan that blows a laminar airflow through this apparatus. This is the radiant panel. Generates a radiant heat source of about 50 kilowatts per square meter, which is pretty intense. You literally have to perform the test with welding goggles on because it's so intense. But what happens is that the samples are loaded on a pan that's placed down here inside the wind tunnel, And on the downwind side is a small coiled wire igniter. So the sample is heated up, all the fuel is pyrolyzed, and the pyrolysis products are carried over that piloted ignition source. So what we did with this is measure the time to ignition for our needles. We saw a few interesting things with radiant only. And again, you know, here we are back looking at moisture content, but what we see from the time to ignition is that for our new needles, the new needles take longer to ignite at the same time of the year than the old needles. But keep in mind, at the scale of the tree, there aren't very many new needles present at that time because the new needle is continuing to develop and elongate. But what we see is they eventually reach about the same level of ignition at the end of by the end of the season. We did see some interesting things that we can't explain yet, and that is that before green up for old needles, we actually have an increase in the time to ignition. So it seems like something that the plant is doing to prepare for the onset of spring in these old needles is actually inhibiting its ignition during springtime, which I think is something we'll probably be looking into pretty soon. So this is one example, but if we plot it against moisture content, we see that during the green up or pre-green up, that period that I showed you that had the high ignition times, we can't explain that variability. But if we remove the pre-green up and only look at when we have new and old needles together, we still, even with a nonlinear model, we can still only explain about 59 to 60% of the variation in time to ignition with moisture content. But we can also explain almost the same amount of variation with either crude fat or non-structural carbohydrates, and the slopes are negative, which means that as the crude fat content of the fuel increases, the time to ignition decreases, which means that they become more flammable. And the same with the non-structural carbohydrates and fats, or the non-structural carbohydrates, it has the same trend. So basically, as the percentage of non-structural carbohydrates increases, the time to ignition decreases. So we have all of these factors that come in together that are very hard to separate. So I guess our solution here is that we should put our fuels on a low-fat, low-carbohydrate diet to make them less flammable. So anyway, but if we look at one of the problems with all of the combustion studies that have been done in live fuels, and I shouldn't say all of them, but a lot of them, is that the heating rate that is used to ignite the sample is very, very slow. In some cases, it takes tens or hundreds of seconds to ignite samples in the lab. Well, that's not very realistic. We know that generally in a wildland fire, the samples are rapidly heating, usually under a mixture of convection and radiation, and a lot of it is convection. So what we did is develop an open flame burner that allows us to simulate a real wildland fire, a real wildland flame, and give us that rapid particle heating that we want. We measured the heat flux at 50 kilowatts per square meter, which is the same heat flux that we get out of the radiant panel, but it turns out that our time to ignition for particles in this apparatus is about 5 times faster. So we can ignite a pine needle in about 8 seconds here, and it takes us 40 seconds with the radiant panel alone. So it's a very neat apparatus, but it presents some new questions. So if we look at our data for this, it's surprisingly not very interesting, because if we look at the new and old needles, we just don't see that big of a variation. Keep in mind that the new needles have a moisture content early on in the season, say almost through August, that's almost double that of the old needles. But what we see is very, very small variations in the time to ignition for new needles versus old needles. And so, but if we look at the difference between the two, we see that in the spring, there's about a 10% difference in the ignition time of old and new needles, despite them having nearly twice as much, twice as high live foliar moisture content. And we do see that as the season progresses, that they come up and approach this one point, which means that the new and old needles ignite in the same amount of time. The big problem is that under this rapid heating environment, live foliar moisture content isn't a very good predictor of the time to ignition. And we'll talk about a couple reasons why. One of the other things that we did just as example tests, this is not very much data to show, but I want to illustrate that when we quench the samples, so we take a pine needle, we slide it out over this open flame burner, it's rapidly heated to ignition, we pull it into that nitrogen environment and we snuff it out. Almost every fire model in the world assumes that all of the moisture has to be evaporated before that particle can ignite. And there's been some other work that suggests that that's probably not right, but what we've found is that at a minimum, most of those particles, even if they start out at 100% moisture content, have about 20% moisture content left at the time of ignition. But for new needles, out here at the high end, we actually had new needles ignite. So they're fully engulfed in flames when they're quenched, and we actually found that they had as much as 130% moisture content left inside them at the point of ignition. So it really poses a question about this balance between moisture and ignition and chemistry and what's happening. One of the other things that I did just as an example to try to see what was going on is using a WFDS, which is a computational fluid dynamics based fire simulator. I recreated the burner that we use, our open flame burner, and added in a particle clump above the burner to look at its heating. But what I did in this case was simulate only the changes in the foliar density. So the density that I showed you earlier, that time series plot where they're almost twice as different, the one on this red plot is the typical density of a new needle, whereas the one on the right, the blue plot, is the typical density of wood that everyone would use as the standard. What you see is a pronounced difference in the time, both the time to ignition and the time that it takes to reach the peak heat release rate. So these are several seconds difference. But you'll notice that the times here are roughly about the times that we were getting from our burner itself during the ignition time from our burner itself. So one of the things that you have to emphasize is that obviously moisture content is important to some extent, but the chemical changes in the plant are changing all sorts of other thermochemical properties that are affecting how that thing burns. And the density changes, which have been virtually completely ignored, could have pronounced effects on its ignition. So if we try to put it all together, the first take-home message here for sure is that when you're looking at live fuel moisture, the live part matters, and it matters because the dry weight and the fresh weight change over time. So if you look at the fresh weight changes, we know that we can probably do a pretty good job predicting the fresh weight changes using the relative water content or maybe leaf water potential. So that's usually a pretty good metric of the plant water status. The dry weight changes themselves, we know that they're mostly related to photosynthesis and the carbon allocation, but also to the phenology and the development of a needle or a leaf over time. So you have non-structural carbohydrates and fats that are the main drivers of the dry weight, and then the relative water content or some other metric can be the main driver of the fresh weight. And it really helps you to kind of decompose what's what's happening inside the plant. So it seems like that a good measure in the beginning would just be to start measuring live moisture content along with relative water content, which is a very simple thing to do, but they tell you something totally different about what's going on inside the plant. And if we look at, but if we put it together in a bigger perspective, we know that the fuel chemistry, so those seasonal changes in all those chemical components have big drivers in a lot of things that affect the particles' flammability, both at the individual needle scale and all the way up to trees and stands. The fuel chemistry affects the particle density and the thermal properties, but it also has an influence on the heat content of the fuel, that again showing you the crude fat linkage, but also the actual moisture content plays a role in determining the fuel moisture, but it also affects this sort of effective heat yield at the scale of the fire, because we know that moisture driven off into a flame makes that flame less efficient. And then so we know that fuel moisture itself and all of its thermal and physical properties affect that fuel's ignitability. So I guess the main take home is that it's that all of these factors kind of come together to form the flammability of this fuel, and to really kind of drive what whether or not it'll burn, and then how it'll burn. So one of the things that I think is interesting to think about is how does the particle ignite, and what's driven off at what time, is because we know that when you start heating up a particle, that you're driving off water. You can see the water go off as you heat it up, either in the lab or in the field. You can see it at the scale of a real fire. And we know that the fuel temperatures are rising over that time. Well, I always kind of relate it to the way that the human body works, because when you eat things, the things that are the most readily available for your body to burn are the simple carbohydrates. So if those simple carbohydrates are the thing that provide the initial pulse to create the flame, and that flame is sustained later on by the pyrolysis of the cellulose, lignin, and hemicellulose, which takes more energy to break those bonds, then you have this sort of staggered basketball effect of each one of these things contributing to the least combustible mixture of the flame at any given time. And we know that the fat content is a little different because it has such a much higher heat content than the carbohydrates. So both the crude fat and the carbohydrates are maybe the things that are providing that initial pulse for the flame. The one thing that's really important is this concept of the radial location. So a lot of models assume that the particle is called what they call thermally thin, which means that it has a uniform temperature throughout the particle. Well, what happens when you heat something up really, really fast is you develop a gradient between the surface and the center of that particle. What's probably happening with the live fuels is that since they're probably not thermally thin, and therefore the only thing that you actually have to heat up to ignition is the surface of that fuel. So basically the ring around the needle or the surface of a leaf, because that's where the initial part of that flame comes from, and then as heat's transferred into the center, more fuel's pyrolyzed. So it's really kind of an interesting thing, because there is no real smoking gun, if you want to call it that, because each one of these factors is interactive, and it influences something else. So we know that the chemistry at the core is a big driver in the dynamics of what we measure as live fuel moisture, but it's also important in driving the thermal and physical properties of the fuel, and also affecting things like the fuel's heat content. So, but for just general, that the higher percentage of conifer needles is allocated to non-fiber carbohydrates than fiber compounds. I think that it's really important for us to know that live fuels are not wood. They're chemically almost completely opposite from wood, and we can't treat them like wood when we're trying to figure out how they burn. and we can't use the thermal and physical properties of wood to reflect that. The second point is that the seasonal variations that we found in live fuel moisture content are mostly related to the dry weight changes. We can explain way more of the differences with the chemistry than, say, relative water content. But relative water content itself can explain some portion of that variation, which is good because it gives us the potential to say that we could separate out that change in fresh weight from the change in dry weight and really see what's happening to the water stress of the plant over time versus what's happening to the chemistry over time. So, and then the last thing is just everything is correlated. The big question is what is burning and when is it burning? So when we're talking about all these different compounds, what's producing that least combustible mixture, and then what's decomposing first to provide that initial source of ignition, because that's most likely what's driving the differences in flammability over time. Okay, and I think that's the end of that, so we can actually take lots of questions now if you're interested.
Well, I certainly appreciate the presentation. I certainly point out to folks that we've had a very good audience today. There is one question outstanding at this point, so we'll go ahead and ask that. I have several of my own. But Sherry Leith asks if you think the relationships will be reflected in herbaceous and woody shrubs in the grasslands as well.
They absolutely do, and we actually already have data, at least some data to show that. We did some work we did some of the sampling in sawgrass actually in the Everglades, and so we have chemistry for basically fresh new sawgrass and old sawgrass, and the relationships are very much the same. This summer we just finished a round of sampling with sagebrush, and the sagebrush is very much the same as well. So we can actually, the same logic that we applied for predicting live fuel moisture in lodgepole pine, we can use identical logic and predict, say, the moisture content of sagebrush with even better accuracy. So I think that it'll probably help.
And you see Gary has asked a question?
I did. It says increasing crude fat increases cutin buildup on the outside of the needle. Therefore, the time to ignition of the needle can ignite can be longer. True or false? And I think that. So here's what I think, Gary, is that the reason that there's a cuticle on a needle is to limit the cuticular conductance. So, and that just means that water can actually diffuse out of a needle through the cuticle. So the plant actually slaps that wax over it to seal up those pores, if you will, and only leave open the stomata. Well, if you burn off of that wax, what you're doing is creating a way for all the moisture that you're heating up and trying to drive off inside the plant, you're giving it a way to get out. So it seems to me like a two-step process where you have to actually open up the pathways to get the water out, and then the water is driven out and then the particle is heated up. The reason I see this is because in those new needles that don't have a cuticle yet, they actually, you don't see the same dynamic basically, but they ignite faster than the old needles from the same place. So when we heat up an old needle, we can actually see the wax on the surface heat up, bubble up, and then evaporate off, and then a few seconds later it will actually ignite. But the new needles will actually ignite. You never see that in new needles, and they ignite faster. So I do think it matters. So BJ, how are you, BJ? Haven't seen you in a while. It seems logical then that a similar effect could relate the difficulty in extinguishment of humid climate fuels to dry climate fuels. Yeah, it does, because the humid climate fuels would be, again, you know, they're not only going to have a higher moisture content, but I always think of it like a quality and quantity thing. So the quality of the foliage is the quality of the foliage actually also determines its flammability. So in a humid climate, it actually has a, one of the things I showed was the heat content for tropical foliage. It actually has lower heat content. So, but I do think that, when the humid climate actually can have a higher relative water content, and therefore actually have more moisture in the fuel, so it should reduce it should actually impede its flammability. But all these things are so interrelated. It's like I find it very difficult to. And we got another question from Gary about sampling brush models for basically needle drape and other decadent loading of dead fuels. I think that's where this is all going to head. Is once we, right now we deal with the sort of particle or leaf level, and that if we have, eventually, once we know things like the phenology of the tree and how that relates up to the whole tree level, that's kind of our next goal, is to say, now we have, at the plant level, we have maybe some new foliage, we have some old foliage, and we have some decadent dead foliage that's mixed in. How does that affect its flammability? So yeah, I'm kind of hoping that that'll be the case. So John, if you'll drop me an email, I'll send you this PowerPoint. My email?
Well, Matt, let me just ask this question. Is this something that is shareable? We could post it on our page that is background information for the webinar as well.
Yeah, some of it's not published yet, so
that's what I was thinking. Yeah.
But I do have, at least the chemistry fuel moisture paper is done, ready to submit, so I should be able to share that with you too. But frankly, I don't care. If you want to post the PowerPoint, I'm happy to share it. Just if we put in the caveat that yeah, some of these things are not published, but the intention is to have them published very soon.
Well, let's leave it that if folks have a particular need, they should get a hold of you and let you know what that need is. And we'll make sure your email address is available as well.
Actually, I can type it in right here if anybody wants it.
Sure. Well, I guess the one thing that I come back to is, we've talked about several things. The fact that we have typically measured live fuel moisture. You've mentioned that there are a couple of things that are perhaps more significant factors, like the relative moisture and the actual fat content. What would you recommend we monitor or try to track? And is there information we can share to learn how to do that? I
think right now the most likely thing to monitor is the relative water content in concert with the live moisture content, with the live foliar moisture content calculated on a dry weight basis, because it'll give you because the one thing that actually Brad just brought it up, but I forgot to mention it. Sometimes it's good to know which audience we're talking to, is this concept of your spring dip. Well, what the spring dip very well could just be an accumulation of carbon in the needles. In fact, there's papers from the 70s that have talked about it. But there may be superimposed in that some water stress as well. We just don't know. But I think that if you had live moisture content and relative water content at the same time, then we would be able to then you might be able to separate those two things out a little better.
Well, so is there a publication or a reference that I can get from you or someone that details the process for estimating these other things?
Yeah, I think we already have all the methods written up for the relative water content. It's ridiculously easy. It's just an extra step. You basically sample the fuel, you weigh it fresh, you dunk it in water and let it hydrate, and then you take it out and blot it off and weigh it again, and that becomes what they call a turgid weight. And that turgid weight is basically the weight of the needle at full saturation. So, and then the calculation is just one extra step. So,
well, I'll make sure that we get that information out for folks that might be interested in doing that. You've got a couple of other, at least one other question here. In the semi-arid juniper communities?
Yeah, I think that that for the longer-term moisture fluctuations, that a lot of it is that we still don't understand how the water status at the whole plant level affects its flammability. So you think about a prolonged drought, then what's really going to happen at the plant level is that, say, you know, its water potential is going to decrease. So it's really trying hard to pull water up out of the soil, and but we don't really understand how the water potential of the plant affects that overall flammability, and I think that's where the longer term moisture fluctuations would come in as defining stress. And definitely in all those semi-arid communities, they're going to be, every year they're going to have stressors, but the thing that's really important to note is very rarely does that water stress correspond to a decrease in the foliar moisture content, for example. There's lots of places where it does work, and in fact we found for sagebrush that the relative water content was almost a perfect predictor of the foliar moisture content, but the chemistry was also just as important. So I think that biome by biome, that you'll find that each of these factors kind of comes in and plays into a different part of the moisture content calculations and how we actually quantify stress at the plant level.
Well, I appreciate that. We have only a few moments left. I will leave the webinar open for a period of time to let people continue to type in some questions that occur to them, and we will make a point of getting answers to those as best we can. You know, I'm sort of with Brad. As I think about this, I think about the spring dip we experience here in the Lake States, that we have a long history of measuring live fuel moistures, foliar needle moistures, and have seen that spring dip year after year after year. And, you know, when it bottoms out is actually after our spring season, which many of our historic fires occur in early May, and that dip reaches its minimum levels actually more in June. And so, you know, it brings back the notion that perhaps, in fact, these needle moistures and their conditions may not drive crown fire in the spring here nearly as much as the dead fuels and the intensities that those surface fires themselves generate.
Yeah, I think it's kind of a trade-off, you know, but you can't discount that it has some impact, particularly at the scale of the whole fire. We know that, you know, energetically, that water it changes everything when you, particularly when you heat it off and drive it off into a flame. So it's just a challenge to separate out all the pieces that we know, all the moving parts. So.
Well, I certainly appreciate it. It looks to me like what's left here are mainly thanks. So I think we'll go ahead and begin the closeout process. Jen, any last comments?
Well, I just wanted to let folks know that we'll do our best to get everything, and I'm sure Listed Lake States as well, recordings posted and all the additional information about the webinar. Please check back with us at our website and our previous events pages. Remember, you can sign up for announcements and everything else. And on the screen are just a few examples of some of the more recent videos through webinars or other presentations that we have available. So check in and please feel free to ask us any questions.
Well, appreciate your sharing the word as well as us for Matt's presentation, Jennifer. I guess I will point out again that if there are questions that folks haven't had a chance to ask, please do type it into the chat box, and I will continue to monitor that, collect those yet this afternoon. I do want to mention that we have monthly webinars here in the Lake States. That next month, Doug Shinneman is going to talk about the interactions of disturbances in using the Border Lakes region, Minnesota and Ontario, where we had blowdown and large fire events occur over the last several years. He's going to talk about that process in a general way and the modeling of that for an understanding of how the landscapes actually interact. That's scheduled for the third Thursday again in January. And we will certainly get word out. If you're on our mailing list, you'll know about that, and we certainly welcome you to sign up and tell your friends about us and join us as you wish. With that, Matt, I really do appreciate the time you've taken. That, like the others have stated, we come into this with preconceived notions, and perhaps we've come out with a little more confusion. But maybe some marching orders to explore some of the new ways of looking at live vegetation. Is there anything else I should say to close, Matt?
No, I just want to say thanks to everybody that came out today, and you have my email address if you're interested in talking more, and hopefully we can all sort through all this together. I find it very interesting myself, and it's always great to have interested partners out there where we can explore together. I'd really like to look at some of the spring dip stuff in the Lake States in the future, and I think we could sort through quite a lot of it if anyone's interested in kind of teaming up. So,
well, I could certainly identify some folks that would be interested in something if there were some data to be collected here I guess I will contact you later to find out what it is I should be looking for and who would be in the best position to help.
Awesome.
I know there's interest here. So with that, I thank everybody for joining us. The last question I see is, are you going to be at the 4th Fire Behavior and Fuels Conference in Raleigh, Matt?
I am planning on it, and then hopefully they'll let me give this there as a talk, as an oral presentation. If not, they actually downgraded this presentation to a poster, which is really kind of curious to me. So I submitted it to present it there in Raleigh, but hope it'll be either a talk or a poster.
All right, well, thank you all. Oh, I have the years wrong. It will be actually next year in January when we give the next webinar. Thanks, Tom. And with that, I'll close the webinar. Have a good day.
Hey, Matt, thanks much. Appreciate it.